So, welcome everyone. This is the last webinar of this semester. I'm very glad to see you all here again. So today we are going to have the talk from Dr. Jafari, sorry, and sometimes I made mistakes for the names. So Jafari is now a PI at both Helsinki University and Tampere University. I think he got this position at Advanced Institute of Research at Tampere University last year. So his group is focusing on developing and applying a systems pharmacology approach for rational combinational therapy in cancer treatment. And today he will talk about how to use this combined therapy for cancer. So the stage is yours. We are looking forward to your talk.
Okay, thank you. Thank you for invitation and hello everyone. And today I'm going to talk about the system pharmacology focusing on combinatorial therapy. So my talk would be in the three parts or three sections. First, briefly, I discuss why we need to do combinatorial therapy. Then I will explain about my approach to choose best combination of the drug. And more broadly, in the last section, I will explain about mechanism of action of combinotherapy, and I will introduce, actually recently we introduced a new mechanism of combinotherapy, which called conjunctional targeting. So our disease model are leukemia, acute leukemia, AML, and ALL. And similar to other disease, other complex disease and cancer, these hematological cancer are very heterogeneous. And this heterogeneity comes from different aspects. It comes from morphology, immunophenotypes, and epigenetic, and also more important on response to treatments. And as an example, based on the FIM AML cohort study, you can see that here they collected 125 patients, but there are more than 200 genes with mutated genes filtered as a high impact mutations on those patients. So on average, each patient has two new or two distinct gene mutations. And if you check the ALL from the FIM cohort study, so the situation is even worse. So for 50 patients, we have more than 700 different mutations. So it means the heterogeneity from the different level of biology, you can follow it. So in this kind of situation, when we have a different, in this kind of complex disease, I can say that as a nature course of treatments that to go for combinatorial therapy. So if you go back in the history, we can say that the combinatorial therapy comes from the sequential combinatorial therapy. So as an example, so one drug, drug A, works very well for many patients, but a large portion of patients have a relapse. And then in the clinics, they try to use another drug. And if the large portion have a good result, then in the future protocol, they will try to combine those drugs together. So this idea it comes from the clinics. But later on, after we have some more information about the biology and background, molecular biology background, then we try to design the complex therapy based on the based on targeting the signaling pathways. So for the case of AML, we have a lots of combination at the level of clinical trial or at the level of clinics, as you can see some example here in this list. And if you check the the regulatory for the clinical trial of the combination therapy, it's larger and too much different from the single drugs for clinical therapy. So this statistic is a little bit old, but it shows that there is a difference and there is a trend to check the combinations. So But if you want to combine different drugs together, you have a difficulty because we have a lots of drugs and the combination exponentially exploded, and you cannot test all of the combination. Let's say only combination of the two drugs. If you go for more combination, the situation is worse. Previously, we introduced a method we called nominal data mining. So one of the application of this method is related to combinatorial therapy, and we try to show and explain here that how we can use nominal data mining for combinatorial therapy. So if you would like to access to this workflow, it is available in our package called NEMA, and also there's a the manuscript is under review, and you can access to the bioarchive. So we designed to have four different studies so far. So one of them is a pilot study that we try to use some public data set contains single drug response information or data, and then we try to use those and test our workflow to suggest the best combination of the drugs. So in this pilot study, we use beta EML as a main study, and also GDSC and allomonoic data sets. So allomonoic data set has both single and combinatorial drug sensitivity assay, but beta EML and GDSC only has the single drugs. So the idea comes, uh, this pilot study was published in Nature Communication, but the idea comes from this assumption that if you want to suggest the best combination, there are different approach. There are, for example, there are focusing on genetic level, genomics level, or transcriptomics level, or gene expression levels, and some other markers. But we prefer to focus on the level of the data which is more close to the phenotype of the disease, phenotype of the patient. And here we have I mean, at least in the in this pilot study and also in FIM cohort data, we have access to the drug response at a, I can say, the large enough um, data size, and then we try to use those drug response. So all of the workflow is started from the drug response level, which is more close to the phenotypes compared to the molecular. But in the later step, we try to validate our analysis using the biological information. So briefly, the overview of this workflow starts from the pre-processing of the drug response data. Then, using the nominal data mining, we reconstruct the weighted bipartite networks between samples or patient samples and the drugs. And then we use the network projection methods to have two different networks, similar drug similarity network and patient similarity network. And after that, we're focusing on analysis of the networks and, for example, extracting the cluster in different similarity networks. And then we check the homogeneity of the cluster using intra-cluster homogeneity analysis using different molecular or biological annotation and information. And after that, when we make sure about our cluster, then we tested in this pilot study the idea of combination of the intercluster between the drugs of the different cluster. So combination of the different cluster together and see how is the synergy analysis. So briefly, this is the bipartite network, and then this is distribution of the weight, which is related to the cell viability. And then we projected, as I mentioned, two different similarity networks, and then do clustering analysis or module finding analysis. And then we check the intra-cluster homogeneity analysis using different level of data. So we show that here that our cluster, which are represented here with the orange and purple box plots, are significantly different from the random groupings from the different levels. So some of these plots related to beta ML data set, and also we repeated in GDSC, so two different data sets, and we show that this approach is not data independent. And also we use similar approach for the patient similarity network just to check that how is the whole workflow works. And after that, we test the intercluster combination to make sure if we have the idea is behind that if we use different drugs from different cluster, then we can target different pathways. So then it would be better to combine drugs from different cluster. And here we showed that with the experiment and also testing in allogeneic data set in the panel A, and also our data set in different cell lines, we showed that if the positive group is related to the intercluster, we showed that if we combine drugs from different cluster, we have a better synergy in respect of different synergy methods, synergy calculating methods, compared to the intra-cluster combination. So that's our pilot study, and then we focus on the FIM-AML datasets. In this work, after our pilot study, we decided to not only focusing on the efficacy of the drugs, but also considering the toxicity. Since we have some drug sensitivity assay related to the healthy individuals in these data sets, then we're focusing on the healthy samples. This work is published in Oncogenetics journal, and we have a similar workflow, but we included more experimental works and different validation methods to make sure about the efficacy and toxicity at the same time. So first we make sure about the clustering after we do the nominal data mining and extract the cluster in drug similarity network. We show that the protein target of the drugs in each cluster are totally different and totally related to two different set of pathways, as you can see in these plots. And then focusing on the toxicity. So here in this plot, we are focusing on this part of drugs. So this is a spot related to the single efficacy of the single drug and toxicity. So toxicity at the y-axis and the efficacy on the x-axis. And we are focusing on this part, the blue dots, because these parts, they have a high efficacy but low toxicity compared to the other dots here. And then we continue to test this combination, and we tested again this time if we consider intercluster compared to the intracluster, do we have a similar result or not? And we show that from a different method of calculating synergy, again, intercluster have a better efficacy, and also interestingly, interestingly, that it has a lower toxicity compared to the intracluster or single drugs. So after that, we use different flow cytometry methods to make sure from our five top combination that we have choose in the previous methods. And then we tested on different patient samples, 5 different patient samples. And then finally, we selected two combinations that you can see the flow cytometry plots here, that in the case of combination, the toxicity, which is related to the cell viability of lymphocytes, it's lower or than compared to the blood cells, and the blood cells, it has a efficacy compared to the different single drug, and you can see that the toxicity is not too much. Again, in another combination that we have proposed, we have a similar behavior. And in addition to that, we also tested how is our flow cytometry correlated with CTG analyzed, and we have a good correlations in both drugs. So finally, we suggested two combinations for AML, two new combination, which we called as a ruxolitinib, olexartinib, or RU, and LY39120 and sapanisertib that we call as LS. So in the last part of this study, we just want to show that how is our new combination works compared to the venetoclax combination, which is some of frontline combination in AML, and we show that in the case of blast cells, they are working similar, but toxicity is much less than venetoclax combination. So then we go for further um, validation using in vivo models and also more ex vivo models. And then we use these works also published in biomedicine and pharmacotherapy journals. And here we use the zebrafish as an in vivo model, so xenograft zebrafish. Then we test our combination. Also, we use RTG to check that this effect of cell that um, we observed at a CTG level would be related to the time course or some how we can see that the behavior is decreasing or not. And we using the different 10 different cell lines, we show that this combination that we have suggested are working better than at least some venetoclax combinations in the time course study. So we can see that this decreasing it's meaningful, and it's not only after, you know, that CTGs read out after only 72 hours, but with RTG we would like to follow up during the times. And also checking the mutation background, and we found based on this study that some mutations are related to this combination. And finally, we go further with some specific patterns. You know that there is a study that AML related to mitochondria, so we call mito high and mito low AMLs, and we show that our combination are worked well in mito high, at least one of our combination RU. And then we use the different cell lines to inject human cell line inject to the zebrafish embryo and test in this two combination compared to the venetoclax combinations at the in vivo models. And you can see here that LS and RU works very well compared to the control and also venetoclax combination. And here is the statistical analysis of our suggested combination. So we also test not only based on the image analysis of the zebrafish, we also using the PCR to make sure that the decreasing, or I mean in the case of RU and LS compared to the different human housekeeping genes, and we show that yes, these two combinations works very well at the molecular level at PCR. Now we are focusing on ALL. So my PhD student focusing on ALL from the FIM institutes. So we have a different size of the data about ALL. So we have 600 drugs similar to AML, but only 28 patients. So then we are focusing again to use the NEMA pipeline to extract the best combinations, as you can see here. And you can see that after we clustering, we even this clustering is only based on the cell viability and based on the drug sensitivity assay level of data, but there are many well matched with the our biological annotation related to the signaling pathways. And we are focusing now on some imputation methods, since our data is not very dense in case of ALL. So we want to make sure about our selection and go for in vitro and in vivo analysis. But after this kind of study, especially after we make sure that our suggested combination are effective more than single and also less toxic. So the mechanism of action of this combination would it was very interesting for us why we have a more effective but less toxic. So then we focus on some experimental methods to extract and deconvolute the target of the drugs. So we developed a method we called CoPISA. If you are familiar with thermal proton profiling, there's a we can say somehow new method from 2013 to explore the target of the small compound protein target, a small compound, using the thermal proton profiling. I mean, after changing the temperature and following up the solubility of the protein, we can shortlist the potential target of the compound. And there is a TPP method and PISA method for the single drug, and we developed that PISA method for the combination, and we called CoPISA. So some of the results I show in the last section are published, actually accepted. It's funny, 26th of February of 26, and we will publish soon. And some of them are not published yet, but this method is developed for the combinatorial therapy. So if I want to explain about the experimental design briefly, so we use the four different cell lines and two different drug combination, RU and LS, and then three replicate, and finally we run 16 TMT runs using 12 fractions. So that's somehow huge proteomics study, which 192 sample run mass spectrometry runs and more than 2000 signals based on the label that we have used for TMT. So as I mentioned for the TPP and PISA, so the assumption is if there is interaction between the proteins and the compound, then the aggregation or solubility behavior of the protein would be changed compared to the control, would be decreased or increased, destabilized or stabilized after there is an interaction between the compound and the proteins. So this is a basic of the PISA methods, that if you use any drugs, then you can see different patterns. But in the case of CoPISA, we follow up these kind of condition, that if we use A as a drug A and drug B, and then we use A, B as a combination, then what would be happen for the targets? And more interestingly, we are interested to follow up the target for AB. So we guess that it would be happen that some targets only targeted in case of combination, but not in case of single drugs. And if that happens, then we call as a conjunctional targeting. If you're familiar with logical gates, so we have an AND gate and OR gate. And in the case of AND gate, we need both compound at the same time, or both agent at the same time to do a function in, I mean, in the general Boolean rules. But in this case, if you have two drugs or two compounds, and if there are some target only targeted in the case of combination, then it means there is an AND gate behavior to target some proteins. But so far we know that, I mean, before this study, we know that the basic of the combinator therapy is to use OR gate, because some proteins, some drugs can target some protein in some pathway, and the other drugs could be target some other proteins, and the together there is a synergy between them, because we can block different pathways, and there is no way for cells to be alive, and we can kill the cancer cells. So we know about those kind of functions for this synergy of the drugs, but the AND gate was our assumption that we want to check in this study. And when we go to our findings, we found that yes, there is some proteins are targeted only in the case of combination. So we use living cells and also cell lysats, two different samples for each combination, RU and LS. And in all cases, we have seen that there are some target specific for the combination, some target for the drug A, some target for drug B, but also there is some common target between these three treatments compared to the control, but there are some specific target only in the case of combination. So here in this offset plot, we can see that, for example, in this case, we have 89 potential targets only in case of combination. That would be very interesting to see what is the function of those proteins and how they affect, and important in the case of AML. And also for other combinations and in living cells and in cell lysats. To test and make sure that so we have some common protein between combination and single drugs, And here is the FC-FC plot, fold change to fold change plot. And we want to show that the change of solubility would be similar or different. So we assume that if there is an interaction between some drugs and the combination, then the solubility would be in the same direction. And here we can see that in all cases, more than 90% correlation shows that the combinations, I mean the proteins that change the protein that has changed the solubility because of the interactions in the case of combination and singles are in the same line, and they have a correlation. So then we do some other analysis with other combination, which this study is not published, but you can see here, this is a FC to FC, not only based on the common protein, the whole protein, and you whole identified protein. And here you can also see that there are some specific proteins that are targeted in case of combinations compared to the single drug. Even we have some common protein target, but there are specific protein also here. There's another study that we try to integrate our method, CoPISA method, with expression data sets. And in this case, also we can see, so the condition for is the case of combination. And you can see that some proteins are even targeted, and the expression of those proteins also different compared to the single drugs. And it mentioned that the mechanism of the combination is totally different, even change the expression, not only related to the solubility change or solubility shifts, is also related to the expression of the proteins. And also, this is another study that we would like to check, that how is the long-term CoPISA and short-term CoPISA. So in the so far, before these plots, we used a short-term CoPISA. So we treat the cells for 15 minutes, and after that we try to check the what happens of the change of the solubility. But in the long term, we use the similar method, but after 5 days. So to enrich DTP cells, drug tolerant persister cells, and then test the solubility change. And in this case also we see that the change of the solubility, even they are not related to expression, so the color here is related to the expression, and also we can see some specific for the combinations. So at the final steps of this part, we try to check that if we combine two drugs together, it might have some non-covalent interaction or covalent reaction between them. Then we have a third molecules. Then all of this story come from the third molecule interaction. But we tested in the mass spectrometry analysis only on the combination, and we have seen that we have only two peaks and related to two drugs, and there is no any third peaks related to the interaction of these two drugs together. So we don't have any interaction, or there is no drug-drug interaction, at least based on this study, based on these methods. So in the later step, we go for the functional analysis and using the AML-related signaling networks. So we made, first we test the different paths related to the AND gate and OR gate. So we show that the AND gate, in the case of living cells and also cell extracts, and the common pathway between them are different totally from the AND gate. So the AND gate and OR gate are totally different, and it means that there are different pathways and there are different logic to affect cells. And we made the AML-related signaling network, so we integrate all of the pathways that we have enriched, and then we extract all of the mutation related to AML, and all of the known target of the drugs related to AML, and then extract all of pair shortest path between those inside that networks. And then we have a AML related network. Then we did clustering analysis and try to do more enrichment analysis based on the biological pathways. And then we found very important, as you can see, some of them we try to show here, are very important proteins which are targeted only in the case of the combination, and they are also important in these networks, and such as NPM1, DNMT3. So those are very important genes that targeted only in the case of combination, not in the case of single drugs. Also, we focus on the kinome tree, and we saw that there are two different kinome profile in the case of combinations and the single drugs, and also these two combinations are a little bit different in the case of kinome profile. And you can see, for example, in the case of AML here, that 20 proteins related only to combinations of the RU, 16 for LS. Those are specific in the AML signaling network, and they have roles for the AML disease. Then the efficacy of the combination could be related to these proteins that we try to show it in the paper. And in the later step, we go for the PTM analysis, and we try to make sure that if that the PTM would change the structure of the protein, and when the compound wants to interact with the protein, then the PTM would affect this interaction. And we would like to check in this analysis that is there any difference in the PTM between common protein identified in the combination and the single drug. And we also found, yes, there is a different PTMs pattern, specifically for some very important proteins such as NPM1 that I have mentioned, and also BLM. So here we also mentioned that the effect of PTM. In another study that is not published yet, we focus on phosphoproteomics. And here also the plot in the middle is related to the combination. And at the level of peptides, we analyze and make sure that there is a different pattern of peptides affected by the combination compared to the single drugs, as you can see in these three volcano plots. And also there's F2 to Fc, or fold change to fold change, for the peptide level, and you can see that there's some specific for the combination in the red dots compared to the single drug. And so one of those proteins, as I provided here, for example, protein H15. So you can see that there is no change in the protein abundance in all condition, control, drug 1, drug 2, and the combination. And also there is no change in the peptide, which there is no PTM of them. But when you focus on the peptide, which has a, in this case, phosphorylation, we can see that in the case of combinations, we have a different peptide abundance. And it mentions that the combination is not only the sum of two drugs, it's a new mechanism that we call as a conjunctional targeting. So last part, I want to discuss about our web tools we call as DORSA. So simultaneously to our web experimental wet labs, we also collected all of the data related to TPP and PISA methods, including our data sets, and collected as a web tools in DORSA web tools. And you can see that many study here, and this part is related to our study, but the others related to the other famous and pioneer work related to TPP and PISA. So if you are looking for some drugs and you want to see is there any target related to that drugs, and you want to see the mechanism of those drugs, it would be good to check these websites to make sure what is the protein target of those drugs, and it would be some clue to continue your analysis and design your experiment. Okay, so as a last slide, I would like to summarize my talks to 3 points. So first, we introduced nominal data mining, which is represented in NEMO-R package. And there's several applications that you can check. And one of them is to propose the combinatorial therapy. Recently, we have also showed that this method would be also good to compare different proteomic search engine. So maybe in the future I will represent it. But the main application that I explained here is related to suggesting the combinatorial therapy. So as a second point, we developed a CoPISA method So with this method, we introduce the conjunctional targeting. There are some proteins that are only targeted or um, only in the case of the combinations, and those proteins have a roles in the disease model that we have studied, and that this mechanism would be interesting, and not only in the pharmacology study, also in any case that, for example, in food science, or any case that you compound two different or many different small molecules or drugs to affect some biological systems. That would be interesting to check that there is some specific target for those combination that you cannot follow it in the single drug or single compound. And finally, I would like to again introduce dorsal web tools. So these web tools would be useful for those are interesting to focusing on some drugs, and it could be very helpful for them. Okay, thank you for your consideration, and I'm happy to answer any question if you have.
Okay, thank you, Dr. Jafari. Sorry, again. Yeah, very professional slides, and actually, I have to say, very comprehensive work and very busy slides. Yeah, so now we are going to the QA session. If you any of the audience have any questions, you can either type your questions in your chat, or you can raise your hand, and we will let you open your mic and ask those questions. So maybe I could start first. So first, congratulations on your new publication. I think that's a very nice work. Yeah, so of course I understand that you have very limited time. You cannot go too much to details about this network thing. Could you briefly explain me how exactly you reconstruct the network? For example, you mentioned this patient similarity network, drug similarity network, and also in your last part, you have mentioned this functional interaction network. Could you briefly mention how these networks were constructed?
Yes, let me get back to the
Yeah.
slides. Yes. Okay, so in the data sets, we have patient samples and drug names. So these two variables are nominal variables. So the patient sample has some labels, and drug names are some Campbell ID. So these two are nominal variables. So we were thinking that how we can extract more information from these nominal variables, and also we have some numerical variables that related to the cell viability of the each patient and each drugs. So each
Mhm.
pair of patient and drugs. So then we reconstruct a bipartite network, as you can see here, but weighted bipartite network. The weight is related to the cell viability. And then
Mhm.
we have this kind of network, and which I mean, they are bipartite because one part related to the patient samples, the other part related to drugs, and there is an interaction between them which is related to cell viability. And then we project the network based on this bipartite network. So then we have two unipartite networks in this case. And then there are, for example, in the case of the drug similarity network, the nodes are drugs, and the edge between the nodes are related to the projection methods that is it comes from the multiplication of the all age related to drugs. So we can say that that weight in the similarity network, it's related to the sum of the weights and the similarity between two drugs working on all samples. So then we have a similarity network drug, but this similarity comes only based on the drug sensitivity assay, not based on any other information. And then we focusing on this network for clustering and the rest of the workflow.
So,
So this,
if I understand you correctly, I mean, for this similarity network, you don't have the similarity ages between drugs or between patients. You only have the ages between your across the two modules. I mean, you only have this bipartity network connecting the patients to the drugs based on their profile.
Yes, in bipartite network, it's only based on these two parts.
But ways in
I
the
said
two
there are
networks.
similarity network,
Yeah.
there is a similarity between two drugs, but this similarities comes from the bipartite, not from the structure or from the any other.
Ah, okay. So actually you use okay, use so for the patients, how did you characterize the similarity between patients if you
Like,
already mentioned
completely
it?
similar. So for the patients, also we have a patient similarity network, and the similarity of the patients comes network, and it mentioned that how two patients behave compared to the whole panel of the drugs. And
Okay.
yeah,
It also but I mean but so you measure some data, right? This data probably about the patient's cells viability after drug treatment, or is this
yes.
how you quantify it? Okay.
And
It's
after
not-
that, we check that how patients are similar based on the transcription or I mean transcriptome,
yeah.
other information that we have. But for the clustering and reconstruct this network, we haven't used anything else except the cell viability of.
Okay. Okay. So then a follow-up question is that right. You have this network, and what algorithm did you use to identify the communities, and how did you determine what is the optimum clustering network clustering results for your bipartite network?
So there are several measures to compare different modularity finding algorithm in networks. So
Yeah.
for example, you can use modularity index, silhouette index, and comparing all of these, we identify what is the best method to find the modules.
Okay.
So
Um,
they're
I
not.
hope there are some more questions. I mean, of course, I can ask some more questions, but I also want the give the opportunity to others to ask some more questions. If any of the audience have any questions, please feel free to raise your hand up, or you open your mic and you can go for that directly. So another thing I would like to ask is like, you know, for this AML.
Mhm.
As I understand, of course, in your study, you have measured the blast cells, right? This is a one important indication for the disease. And actually, there's another cells called the neutrophils. That is also used as an indication for the development of cells. So you want to ensure the neutrophils is also not cross certain thresholds. So how was this considered, or was neutrophils also considered in your study for AML? Because I don't remember I have seen the neutrophils, but only the blasts.
So in flow cytometry analysis, we use the different markers for the different group of thing in this. No, no, not this one. This one. So as you can see here, we have a different markers. I
Yeah.
couldn't remember which one related to neutrophil, but our method is only not only but mainly based on the blast cells. So we try
Okay.
to put the best. So there's the top combinations of the drugs based on two different clusters, and we choose the RU and LY mainly based on the blast and also lymphocyte
Lymphocytes. Yeah.
toxicity measures.
So there are no neutrophil data actually here.
I'm not sure about the marker. I couldn't remember
Yeah,
now.
maybe some markers are relevant, but I remember in some studies they emphasize on these neutrophils because this is another indication to control the progressing of the AML. That's what I understand.
We
Yeah.
follow the recommendation of our collaborator, uh,
Mhm.
Caroline Hickman, who is focusing on AML and more focusing.
Yeah. So one more technique question. Could you elaborate how did you identify these both proteins or both drugs are functioning in this AND gate? Is this based on prediction, or this is like um, they have the same target, or how was this characterized for these AND gate proteins? It's
So
like proteins they you show this Boolean gates that you have this combined proteins that functional the like with in a get function and get function. So how was this exactly identified? I don't get it. So
Actually, the AND gate idea comes from the assumption that we have. So
okay,
you see that if this eight case that you can see here, so it might be some target specific for drug A, some target specific for drug B, and if there is some target specific for AB, not for A and B. Only in the
mhm,
case of AB, then it mean we concluded that would be AND gates. There is some common target between A and B, or AB and AB and B, or all three treatments. But these are could be related to OR gate. But if there is a target specific for the combination of treatment of the combination, which is called as AB, those
okay.
targets would be related to AND gate. And I can say, to best of my knowledge, this is a only method that we can follow up the AND gate at the signaling level. So far, our knowledge about the logical gate in biology related to the gene regulation and epigenetic study. But if you want to focus on signaling network and you make sure about I mean, if you want to have some high throughput method to find the AND gate and OR gate at the signaling levels, and that the CoPISA would be the one, I mean, I can say the only method so far that you can identify this behavior. That there are some target only locked in the case of treatment of combination,
So
not
so to clarify this, so when you are talking about these targets of the different treatments, you can have the targets for individual drugs like A and B, right? Or A and B.
yes.
And how do you know the targets for the combined drugs? I mean, my simple understanding is like you have their individual targets, and then you know what will be the combined targets. And how do you know there whether you get new targets for the combined treatment? Is this because some off-target effects? How does this work? I mean, if you combine two drugs, how do they get new targets? I mean, they can only bind to the proteins they know to bind. They will not bind to new proteins. Or that's why, for
So
example, here you have the two circles, three circles, the targets for protein A and the targets for protein B, and then the targets for the combined protein drug A and B, right?
yes,
So my understanding is like the targets for the combined therapy can only be from A or B, but cannot,
that is OR gate.
yeah,
If A or
or
B,
aggregated.
yes,
Yes, but there from your Venn diagram, it seems like there are new targets that are not
exactly.
new targets of the treatment. How does this work for drug? I'm a bit confused here.
But there are some new targets in case of combination that we cannot follow them in the single drugs. So these target those protein, they have a shift in solubility only in case of two drugs, not in case of one drugs or any of
Ah,
them.
okay. These are not based on the physical interactions, but based on your this whatever this profile.
Profiling and
Ah, okay, I that's why. Okay, it's not okay. I see.
so I
Okay,
would like to go further and check, for example, to extract those protein and using
okay,
some biophysical method to make sure about how is the mechanism in the molecular mechanism for the interaction. But based on solubility assays and solubility shift, we found some new target only in the case of combination. And then we conclude those are related to the AND gate.
I see. Okay, that's clear. So I see someone turn on the camera, and is he supposed to make questions? This I don't know. I mean, he doesn't have a name, but he has this mobile name. Maybe, yeah, maybe she just accidentally turned on the camera. So of course, then some more questions, in broad concept, and right. So you have identified these potential combined therapies. I mean, is there a like a follow-up plan to go for clinic trials or some or further studies on that?
So now we are focusing on the combination of the CoPISA with expression proteomics, and
Yeah,
also my student here in Tampere focusing on developing new methods and that we call COPRA. and that one is totally different from the temperature treatment. So we use the different method to identify a specific target. So his preliminary result also show that based on that methods, we also find some specific or new target only in the case of combinations.
Mm.
And at the same time, we are also working on ALL disease. So that's another disease model that we would like to check, and we would like to make sure about this workflow also working on that disease too.
So, I mean, of course, there are also a lot of these AI models to predict new drugs. So, what's your opinion? Whether this kind of approach can be also used for combined drugs, or whether there are enough data to train such a model? Mm.
I
Mm.
one approach for the future would be to for example, after we identify the combination of these two drugs would be better and compared to the single drugs, and if you want to go further to translation this finding to the clinics or to the pharmacological industry, then most of the pharmacological industry would like to design the new molecules. So if there's some methods to design the new drugs that or new compound that could play the role of combination. So any a specific chemical group that would be a substitute in the new drug that would be used as an alternative of the combination. That would be one direction in the future. But so far, I haven't checked it, and just as a
Mm.
idea would be interesting in the future.
Yeah, I see. Okay, so any questions from the audience? I hope someone will be brave to ask questions.
Maybe Alfonso.
So, it seems that's no. Okay.
Mohit,
So
very nice talk.
Alfonso is here. Okay, please go ahead, Alfonso. Yeah.
Mohit, very nice talk. Sorry, I had to drift a little bit while I was listening, but the approach seems to be very good. We should really look into kind of this drug-drug interactions because that's also what we have done, and we have quite a lot of data of combinations of drugs that drug response in cell lines, especially, and also different kind of chemotherapy resistant clones in solid tumors. So I don't have a specific question at the moment, but just kind of I'm launching you the ball to tell you that we should definitely sit down and have a look a little bit more deeply about that.
Okay, thank you. Thank you for your comment.
Sure. Collaboration is always appreciated. If you want to collaborate, I mean, just go for him. And yeah, other questions? Daria, maybe the microphone of Daria is on. Do you want to ask questions, Daria? No, no response. Okay, okay, fine. I think yeah. So I think we like to thank you again, Dr. Jafari. And yeah, that's very nice talk. And we learned a lot about your combined therapy. And also, once again, congratulations for a new publication. And yeah, and we will meet and talk in the faculty and to see whether there are collaboration yeah, for the
Okay.
future. And I also want to thank all the audience for coming. And this will be the last webinar of this semester. And as Yulia has already announced, we have this bioinformatics day in June. So it's free for the participants if you register for the membership of the our society, and our society membership is very cheap. I mean, it's like €6 for students. So and then you can have a really good chance to listen to the good great talks and show your research work by presenting posters or even submit an abstract for getting a talk.
And
So
when is the